Thank you very much for the opportunity to present to you today and for this generous invitation. I sincerely apologize for not being there in person. As you know, travel nowadays is not as trivial as it used to be, especially when it involves Asia. Today, I'm going to talk about something that I'm deeply passionate about, and that's artificial intelligence for drug discovery, but also for longevity. The topic which is very dear to my heart, and I'm pretty sure to all of your hearts as well, because that technically defines how long those hearts are going to beat. I need to make all those legal disclaimers because the company is in a specific mode where we cannot provide forward-looking statements and not investment advice. And I'd like to first ask you to think about what is the biggest problem in human life and in life in general. Well, I've asked myself this question around early 2000s when I finally got to the point where I became financially sustainable in IT and could do pretty much whatever I want. And I realized that the major problem that I'm facing is aging. So the fundamental problem of human life is that we grow, mature, reproduce, take care of our young, reach our peak, and then we decline and die. And there are many ways to extend that. So diet, exercise, sleep, meditation, anything you can think of is going to marginally extend it and maybe reduce the probability of disease. However, we are not made like Blancpain watch. We're not made to last forever, and we don't have a lot of replaceable parts, unfortunately. And at this point in time, we cannot track, repair, and continuously improve. So, and I think that this is a fundamental problem of nature, the lack of the ability to continuously improve regardless of how hard you work. Also, if you think about complete elimination of diseases, this chart does not come from my work. It's 2006 Aging Research Reviews, one of the papers describing what would happen if you completely eliminate a disease in the population. How would it increase the life expectancy in the United States at birth and at age 65? So here are the numbers. Usually, if you ask pretty much anybody on the street, they will tell you, oh, if we completely eliminate cancer, we're going to gain 10, 15 years, maybe 20 years to life expectancy. Well, no. At birth in the US, we would get 2.3. At age 65, 1.2. So it's not cancer that kills us, even though it's a huge problem and needs to be resolved. But there are many other diseases and aging itself. So whatever doesn't kill you in terms of disease, aging will ensure that something else will. So that's why I decided to dedicate pretty much everything that I've got, all of my time. So I don't have hobbies, don't have family, don't plan to leave an inheritance. I spend everything I have on the science of longevity biotechnology, learning how to extend human life using means that go beyond optimization of diet, sleep, and exercise. And we run a conference. I established it in Basel originally and moved to Copenhagen. I highly encourage you to attend the next one, the 10th annual, the largest meeting for aging research and drug discovery, bringing together all the key opinion leaders in this field. And it's also a 5-day ultra credible conference run by the University of Copenhagen and Insilico. And what is the main obstacle for increasing healthy longevity for everyone, in the age of technological progress? And the main problem, the way I see it, is this chart. So if you are not familiar with how drugs are discovered and developed, please do take a picture, and that will be your cheat sheet for pretty much anything pharma. So it starts with disease hypothesis and target discovery. So we need to understand the mechanism of disease, why the disease happens, and then narrow it down to a specific protein, that is called a target that we can perturb using a small molecule, chemical, or a biologic. The process takes 1 to 10 years. Sometimes it may take you, a eureka moment, and you can do it immediately. Usually it's funded by the government. Usually it's done in academic institutions. and it may cost you, billions of dollars to decipher the mechanism of disease. Many diseases we don't know the target, we don't know the mechanism. Like for example, Alzheimer's, we're still struggling to understand why it happens. But once you know the target, you start discovering the drug, looking for a great chemistry or biologic. Here is the time it takes, the probability of success, and then you start human clinical trials, establish safety in phase 1, efficacy in phase 2, and then safety and efficacy in phase 3. And just this part costs you on average about $2 billion. In today's money, it's even more. It was the paper published in 2010. So I decided to focus all my energy originally to utilize AI to accelerate this process and significantly de-risk it. So AI can help, and specifically modern AI, and I'm talking about deep generative reinforcement learning. So the technology that allows you to win Go competitions against the Go masters, StarCraft, figure out new strategies, generate great art, etc. So you can use this technology for biological age prediction and monitoring to develop those aging clocks. Actually, since the Blancpain is one of the sponsors, you can develop a clock which will show your biological age in addition to chronological time. Biomarker development, biological target identification, molecular generation, and also precision medicine. So at Deep Longevity, we focus predominantly on age prediction and monitoring, and also mental health, depression, motivational behavioral modifications. Actually, tomorrow we have a pretty cool paper showing how psychology changes in time with your biology. So you are a different person every time you get older. And Insilico covers the rest of the AI applications for drug discovery. We managed to build a company which is spanning pretty much every time zone. We're one of the first AI-powered drug discovery companies with over 250 scientists globally. We've been recognized by top industry publications. And one of the research papers that we publish many, usually about 1 to 2 a month. But this one is specifically notable because it showed, we did it with Alana Spuragudzik, one of the directors of the Vector Institute in Toronto, before that at Harvard, a famous professor. So we actually became pretty good friends. That's me in his lab this year. So what we showed is that if you give us a target, we can use AI, generative AI, to very rapidly generate small molecule drugs, test them all the way into human cells, and then organoids, and then in mice in 46 days, if you're using a known established target. So this was a pretty popular paper and showed acceleration of drug discovery at the early stages. We then took many concepts in that paper and also in many other papers to come up with a set of software that is now used by 9 of the top 30 pharmaceutical companies that allows you to identify targets using different heterogeneous multi-omics data to formulate the disease hypothesis. So simple that the high school students can use it, and they used it to demonstrate that they can accelerate drug discovery and identify novel targets. They just presented at this conference in Copenhagen. Then you can generate small molecules with the desired properties given the target, and then predict the outcomes of clinical trials. So we try to go end to end. And I had a question in 2020. Can we now use this software that everybody else uses to go ourselves and discover a therapeutic with dual purpose that would target aging pathway, and disease at the same time? And we came up with this goal that is pretty much, it's very rare for a big pharmaceutical company to discover a target, design a molecule, and take it all the way to clinical trials. Usually the most productive ones design and develop and put on the market about 1 a year. And it takes them decades to get there. So we decided to take this challenge, raised a lot of money. So we raised over $415 million to date from healthcare and technology investors, and decided to go after this goal. We identified using pandomics a target which is implicated in several age-associated biological processes like lung fibrosis, skin fibrosis, kidney fibrosis, and associated diseases. nominated preclinical candidates for those diseases. And then very rapidly from scratch, from zero to a preclinical candidate. Preclinical candidate is when you test the molecule in at least 2 in vivo efficacy studies. So mouse models and human xenografts, for example. In this case, it's mouse models, lung-induced, pharmaceutically induced fibrosis in the lung. And we managed to nominate the preclinical candidate in under 18 months on a very small budget. So for IPF, it was under $3 million. Usually in traditional world, it takes you about 4.5 years and about $500 million to get there. And then very rapidly, we went to phase 1 clinical trials. So right now it's in humans, already tested for safety. Fingers crossed, took us under 30 months to get from target discovery to phase 1. So developing a novel therapeutic using AI. So now what about the future? Can we accelerate this process, demonetize it, democratize it? And I started building one of the most sophisticated target discovery labs in Suzhou originally, in China. So we have an AI-powered drug discovery lab. So one of the big problems in biological sciences is reproducibility of the experiments. Sometimes, postdoc or a scientist in the lab screws up, or the data gets is inconsistent. There is laboratory error. So what we are trying to do, we're trying to bet on those AGVs, autonomous guided vehicles that transport microplates, reagents, cells, organoids from station to station. We have multiple single arm and dual arm laboratory robots. As you know, in China, these guys, deliver food in the elevator to you from downstairs. They are very ubiquitous. You can repurpose them for laboratories. So we have compound management system, cell culture management system, high throughput screening system, next generation sequencing system, and also imaging system that are fully automated by themselves. And fully roboticized. So actually humans do not go into this part of the lab where those AGVs are operating. We can work with 3D culture of human cells. We can work with 2D culture. I even took biopsies of my own tissues from multiple tissues and expanded them using iPSC technologies, made organoids out of those tissues. And we also can work with different diseases, predominantly cancer. We can screen a lot of samples at the same time. Usually it's about 500 samples a day using fully autonomous high throughput screening system. It actually doesn't sound like much, 500 samples a day, but we get a lot of information from every sample. We get deep phenotyping, so full imaging for every sample, gene expression and methylation data as well, and also incubate the samples with many compounds. We also have dual arm robotic systems that allow you to do high throughput laboratory experiments, but also do very human-like moves. Pipetting with very high precision and specifically like humans would do it in a laboratory book. And we also have pretty much standard sequencing equipment, but we fully automated it to be able to completely eliminate human error. I'm launching this lab in November, and hopefully we'll be able to scale it and put many of those labs in different countries to accelerate target discovery and understand why disease happens. And also we'll be able to study aging more effectively. So that's me. Again, very dedicated to longevity research. Happy to answer any questions. And thanks to the organizers for inviting me to speak today.